I think the most exciting part of the project is the idea that nobody's done this before. We're figuring out a way to study cells that's quite different from any other device or process to do that.
We hope that our project marks the beginning of research that's really going to help people.
We're Team 7, and we're developing novel devices to study cellular alignment in biomedical devices.
The focus for this project is atrial fibrillation, or AFib, which is a common arrhythmia where the signals that cause the atria of the heart to pump are disorganized. The pathology of AFib is really broad, but one of the biggest risks that comes with it is the risk of clotting and stroke.
Specifically, there's a structure in the heart called the left atrial appendage, and in AFib patients, this structure is highly prone to forming blood clots. So if the patient is not eligible for oral anticoagulants, this structure is sealed off with a device called a Left Atrial Appendage Occlusion, or LAAO device.
But those devices can also cause clots to form, and it's not fully understood how that happens.
The cells that grow over the device can align and change in response to both flow and the surface they're growing on, and that can play a big role in whether a clot will form or not. Can the interaction between the surface of the device and the surrounding flow teach us anything about why blood clotting happens?
So that's the big question. How do flow and surface contribute to cellular alignment?
So right now, as people are studying this, they are using a parallel plate setup. While that does let you gather information, the parallel plate setup is restricted in its ability to three-dimensionally mimic blood vessels and other physiological areas of interest.
We want to study cells on nanofibers under flow conditions, but we want to use devices capable of studying the cell alignment in 3D. We want to actually put these cells in pipes and models of the left atrium so that we can get closer to the conditions you'd see inside the body.
We are building custom models and testing setups to do exactly that.
We started with the series device. Designed to test multiple experimental conditions at the same time, this biocompatible device has 6 identical testing sites. The sites are carefully designed to minimize disruptions to flow inside the pipe, so that the very first site in the row is experiencing the same flow as the last site.
The series device is good for creating simple pipe flow, ideal for early experimentation and altering multiple factors at once. But we also want to have a setup that's closer to biological conditions. Enter the left atrium model device, or the LA device for short. This adapts the culture site design to a patient-specific model, so you can study cell alignment within an actual model of the heart.
We built a modular testing setup that allows us to use either device and multiple different kinds of flow with the cells to fit inside an incubator, and then we put it to work.
Our initial short-term results indicate that both flow and substrate are playing a role in how cells align and behave on nanofiber scaffolds. They also prove that our devices are capable of supporting cells, so they'll enable future studies as well.
Through testing,
I can feel the reservoir. It's beating like a heart. That's kind of neat.
And troubleshooting.
Do you think that's enough tape?
And plenty of late nights in the lab.
We've developed procedures, devices, and testing setups to study cells on nanofiber scaffolds under a variety of flow conditions never tested before.
but we certainly haven't seen it all yet, and future improvements would include more testing with longer test durations and improved instrumentation to better analyze our flow. That would allow us to better mimic biological conditions and make the setup as lifelike as possible.
We'd like to thank our fantastic advisors and TAs, as well as the students and professors who generously donated their time, resources, and expertise to this project.
We're Team 7 from MTU Biomedical Engineering, and we're excited to see where this technology goes next.